So the story I'm going to tell today is kind of the story of my lab, really, and what I've done since starting the lab in 2007. I'll walk through just a series of short little stories about some of the research projects we worked on over the years. And then kind of last half of the talk will focus on the stars of the show, which are these guys, the Chrysochromata parva, eukaryotic algae I work on, and the viruses that infect it. So again, thinking about the beginning, Alice already gave a great introduction, told my life story. I was thinking about where would I start this talk? When I was writing the talk, I thought, what's the starting point? And I really decided I wanted to go all the way back to what got me interested in all of this work in the first place. And I did my undergrad at UBC, as Alice had said. And at that time, kind of right about here, 1990s, I became acquainted with this Keeling curve. I think most of you have seen this, right? Charles Keeling studying atmospheric carbon dioxide concentrations from Mauna Loa at a location free from industrial input in that. And what Keeling had reported to US Congress in that is that anthropogenic emissions were pushing the carbon cycle and forcing the increase of CO2 in the atmosphere. And that was at about this time when I was in my undergrad, when I first heard about this and thought how alarming that was when we were at a nice low value of about 350 parts per million. You look at where we are today, it's way, way higher. The amazing thing about this that struck me is we can actually see the breathing of the planet, right? We can see that when the curve dips down every year, that's spring, summer, increased photosynthesis, drawing down CO2 out of the atmosphere. And then you see fall, winter, that increase in atmospheric CO2 as all that organic material produced is then respired back into the atmosphere. So the measurements are sensitive enough to capture annual cycles of photosynthesis in the northern hemisphere. And yet, are still denied by some people that this is not actually happening. Nonetheless, it's happening. And it got me excited about thinking, well, what are humans doing to the planet and how are we affecting it? I then also started to learn about this carbon cycle. This is a picture. This is a fairly, well, not that recent anymore. This is Climate Change 2013 from IPCC, the International Panel on Climate Change. Really, the story hasn't changed since then. This is a carbon budget for the planet and thinking about where all the anthropogenic emissions are coming from and where that carbon is going. And so we can see atmospheric increases in carbon have been pretty remarkable over the Anthropocene. And then what really interested me was looking at this box here, marine biota. And I thought, that's interesting. There's a pathway for carbon to be flowing out of the atmosphere into the surface waters where it can be sequestered into the deep ocean and maybe even subducted down into the continental margins and lost forever. And what this represents is a pathway of carbon removal that can last for thousands of years or even longer over geological time periods. And this fascinated me to think about the biota of the planet actually driving carbon cycles. And for me, that was a new thing. Now, I think most of us kind of understand that that's happening. I was really curious about what was in that box, what was in that marine biota box. And it's this. This is composite satellite image from CWIF satellite and now defunct satellite. But what you're looking at, the colors are chlorophyll A in the oceans and lakes. And where you see the greens and red and orange, that's where there's a lot of chlorophyll A. And where it's blue and purple, that's where there's very little chlorophyll A. And this is all chlorophyll being detected in microscopic plants or algae, right? And so when I realized that these organisms that are invisible to the naked eye are actually controlling planetary cycles, I was just struck by this. And what I love about looking at this imagery is you can really see how the planet again is breathing, right? Where you see that increase in chlorophyll in spring, summer, and then the respiration. And all of that is really setting how our climate works and controlling climate in a really profound way. And so this I thought was incredibly exciting. Then I got even more excited when I saw these reports. And this is how life can be so serendipitous. I took a psych class and I learned about algal viruses. And that might not sound right, but what it was is that there was a textbook for this psych class I took that had a complimentary magazine on the back of it, a Discover magazine. And in that Discover magazine that I just happened to be flipping through, there was an article about the high abundance of viruses in the marine environment. And at that time, early 90s, that was a relatively new and profound discovery. And that just struck me as, okay, I was interested in carbon cycling and algae and all that, and now there's all these viruses that are infecting them and doing something to the ecology that I want to learn about. And so this report in 1989 came out. There's some interesting science backstory behind that about people being scooped and a little bit of dirty politics and all kinds of things I could get into later. I won't sully this talk with that. But this report by the Norwegian group really showed us that there was viruses in high abundances. We're talking millions to tens or hundreds of millions of particles per milliliter of seawater in every sample they looked at. And that was all based on microscopy like this, where they could just take a seawater sample, put it on an electron microscopy grid, stain it with uranyl acetate, and take a look. And you see all kinds of small virus particles, some larger particles like this one over here, or some of these weird amorphous particles. Point is that every seawater sample you can look at will have millions and millions of viruses. And their question became, what are they infecting? What are they doing? And what activity do they have? What influence do they have on the ecology of the system? Bringing it closer to home, some of you might know Roxanne Maringer, who worked with Dave Bird back in the 90s and did a study of 30 Quebec lakes, and as well some marine samples. And what they found is that no matter where they looked, they also found that concentrations of viruses were about 10-fold higher than the concentration of bacteria in the same sample. And that typically there was about 10-fold higher viral abundances in fresh waters than in the ocean. So again, the aquatic viruses was really being anchored at that time in the early 90s as a really strong new discipline in aquatic ecology and in aquatic microbiology. And then I joined the Subtle Lab to do a PhD, and I was sitting on a boat in the Gulf of Mexico when a postdoc at the time, Steve Wilhelm, who's down at the University of Tennessee, Knoxville, and Curtis were having a conversation about how viruses might be influencing food webs. And they coined this phrase, the viral shunt, that was first published in a bioscience paper where they wrote up this idea. And the idea is pretty simple, really, is that if you have primary production feeding higher trophic levels, ultimately moving carbon to the highest trophic level, that represents your basic flow of energy through a standard food chain as part of a food web. What they imagined, though, is that if viruses were infecting phytoplankton, converting some of that organic material from photosynthesis into particulate and dissolved organic matter pools, that then becomes fodder for heterotrophic bacterial growth, ultimately leading to an increase in respiration in the system and a loss of that primary production from higher trophic levels. So they called this the viral shunt, a way that viruses were shunting primary production away from the top food chain down into microbial food webs, ultimately leading to net respiration. Now, this is only one idea of how this influence can happen. It is borne out in some studies, not in others. Again, it's more of a hypothesis and an idea about what viruses might be doing. Now, fast forward, thousands of papers have been published on this topic over the last year, and a lot of that was summarized quite a while ago by Carina Broussard at NIOS in the Netherlands. And she pointed out to us in this review article that still holds true to this day, that we now have solid evidence that viruses are controlling the diversity and gene flow in aquatic ecosystems. They're controlling nutrient recycling by moving inorganic materials between different pools, and also influencing organic matter cycling. So really, what this all suggests is that viruses are having a profound effect on both the ecology and evolution of aquatic ecosystems, period, full stop. So with all that little bit of background, that's where I decided I wanted to pursue viruses and keep studying them for my career. And so now I'm just going to walk through a few of the early studies that the lab did to try and understand how viruses were influencing freshwater food webs. And so the first thing we did, bit of a low-hanging fruit, pre-metagenomic era, is we just went into freshwater environments. I had some samples from Colorado, Chatfield Reservoir and South Platte, as well from some samples from Ontario. We simply did clone libraries and PCR sequencing of all these samples. And at that time, this was early 2000s, while we did this study in 2007, we thought we had a universal PCR primer set that would amplify virus genes in all of these algoviruses. At that time, the only group of algoviruses that were known or described in the literature were phycotonaviruses, or large double-stranded DNA viruses that infect algae. And we knew the DNA polymerase gene sequence for those, so we developed a universal primer to try and amplify viruses from different environments and try and get a first sense of the diversity of those things. And this is what we got. All of the sequences in red and orange were from Ontario, green and purple were from Colorado. The light blue that you see down here and up at the top represent all of the known cultured algal viruses at that time, so not many. What was really surprising to us about this, not just the diversity, all the different species, these were clustering at a 95% identity at amino acids, so even further apart when you think about nucleic acids. What was stunning to us, though, is that this little group down here in light blue are the only known freshwater algal viruses at that time. And everything we discovered was related to these marine viruses that infect prasinophyte algae. And that's really bizarre because there's almost no known prasinophyte algae that are described as freshwater species. So we still don't have a clue of who the hosts are for all of this diversity, but it did tell us that there was a remarkable diversity of viruses out there in our sites, and it told me that I wasn't lying when I gave my job talk saying that I could detect freshwater viruses in Lake Ontario. So then with that gene sequence in our pocket, we decided we could develop some qPCR methods to track individual viruses and look at their dynamics in the environment. And so we just pulled a few different sequences that we were interested in from a couple of different groups that we had detected, developed TACMAN assays, or 5' nuclease assay, primer and probes. This is borrowing from stuff I did in my postdoc, and just decided to track these viruses over a couple of years of weekly sampling. And what we found is that there was distinct periods where each virus would have a prominent abundance and then drop below down to low levels. And I'll just say with this qPCR, we made sure we did all sorts of validation to make sure that one primer probe was targeting truly one sequence and not anything else. And so the point here was that every virus had a distinct phenology or seasonal timing. Some seemed to persist for long periods. Some were very ephemeral and just appeared for a week or two and then disappeared. Often, though, the viruses never dropped completely below detection. We could always detect remnants or traces of them, which is also very weird because one thing we know about algal dynamics is that most species have a pronounced seasonal phenology themselves, meaning they're present at certain times of the year when you can detect them, and then they disappear from samples for the remaining part of the year, probably encysting or being in the sediments or something like that. We don't really know too much about that, but it got me curious about how do these viruses return year after year after year if their host drops below a density that would support viral replication, right? Viruses, we should all know, are density dependent for the replication. If the host's gone, the virus doesn't last. And at that time, a few studies of viruses' decay rates in natural samples suggested that virus turnover was very high. That viruses are not very good at withstanding environmental insults like UV light, and usually viruses would be destroyed in a matter of days. So I was really curious about how those viruses would come back each year. And it's something I'll return to later in the talk. And just, again, to punctuate this a little bit more, I had a master's student, Robin Roson, who'd worked for the summer with Fisheries and Oceans Canada, sampling a compromised embayment of Lake Ontario, the Bay of Quinte, which was known for a lot of phosphorus mitigation efforts and things like that to prevent eutrophication, or to roll back eutrophication, I should say. And we, again, looked at a bunch of different viruses to determine what their ecology was. And what we were hoping is that viruses that were related to each other would have a similar ecology that would allow us to easily model these things. Yeah, good luck. What a naive idea, right? So when we look at every single virus had its own distinct distribution. The different lines here represent 3 different sites in the Bay of Quinte, and the x-axis is time over one growing season. And what you can see is that some viruses seem pretty stable across the entire Bay of Quinte throughout the entire year, where other ones have really episodic presence and absence. Some are only present at a couple of sites, not all of them. Really what it told us is that every virus, no matter how closely related they were, like for example, these prosinovirus 68, 20, and 49, those are just named after the clones that we'd identify from our sequencing. These are not cultured. But the point is that every one of them behaved quite distinctly. And maybe it was naive to ever think we could bin these things, but clearly we can't. And each one behaves like a snowflake in the environment, which is both sobering and maybe a little bit depressing at times, because it makes studying these very, very difficult. But at the same time, it's really exciting because there's a lot to know. So again, going back to thinking about how do the viruses return year over year, I decided that I'd return to looking at sort of a virus decay rates. How are viruses being destroyed in the environment? How long can they last? And all the studies that I'd seen had focused on single sample times. So you basically take a virus put it into some kind of bottle or reservoir, put it in the environment, and monitor it for a few weeks and watch virus infectivity or gene copies decline. And most of those have been single point studies. So we decided we'd look at different seasons. So we used the UTM stormwater pond. There's all kinds of interesting stories about that as well. But point is, we sampled in spring, summer, fall, and winter. And here we were assaying infectivity for a couple of different chloroviruses. So these are viruses that, fortunately enough, we can grow on solid agar media and do plaque assays to get really accurate counts of their infectivity. We also had this other virus that I'll talk a lot more about. This is my star, Chrysochromata parva, and that we can't grow it on solid media. We've tried many, many different times. It just won't do it. But we can still assay infectivity using a most probable number assay, or using endpoint dilution assays like this in this 96 well plate. When we looked at that, we got some interesting patterns. First, my student, Andrew, had gone out and sampled destructively these bottles and then assayed them for the infectivity at these times of the year. When he went out to get his December samples for the winter, this is what he found. The rack was frozen over, and we actually couldn't get our samples from that. We got the first 2 time points, and then the last 2 time points, which were supposed to be 2 weeks and 4 weeks later. It was not recoverable. So they sat over the winter until the spring when the pond melted and we could go finally recover our bottles. And at that point, he said, I think I'm just going to pour all these out. There's nothing there. I'm not going to worry about it. And I was like, no, it's just, weeks of your life. Go ahead, do those infectivity assays. He was a little angry with me, but in the end, it turned out to be a good guess to do that. So he, after 128 days of having these sit, he did the infectivity assays. And what we did find, which was really surprising to us, is that there was times of the year, like the winter months. So this plot is showing infectivity decay rate on a per hour, percent per hour basis. So meaning something that's decaying at 10% per hour, that's lost in days, right? The half-life of those viruses is very short. And that you can see is pronounced in the summer. But in the winter, what we found is that over 50% of the infectivity remained after 128 days being frozen. And that was surprising to us because these viruses, if we just put them in the freezer, we lose all infectivity. We can't freeze them. But in the environment, I guess under a slow freezing or a slow chill, they can survive that and overwinter. And so that was one answer, that the viruses could actually overwinter in the water column. We've also done a lot of other work now starting to look at sediments, but I won't get into that story. Final point I'll make about this, I should have retired after this. This was the best publication I've ever had because the authorship is long and short. The guy's name was Andrew Long, so it was kind of a nice one. Like I said, I was done at that point. I should have just left it. Okay, so I also had another student, Mike Stanevsky, who wanted to look at the effect of viruses on the whole community of phytoplankton and how viruses maybe had an effect on primary production, but also then boring down into looking at, using our qPCR methods, individual species and seeing how viruses impacted them to see if a community signal was losing a lot of the dynamics you'd see at a population level. And so we borrowed from classic oceanography experiments, Connie, you would know these probably pretty well, the dilution approach to look at grazing rates. And the way this experiment works is you take a whole water sample and then you filter it in a way that removes all the phytoplankton and all the grazers. And you add it back to the whole water at varying proportions. So at the highest dilution levels, you've essentially reduced the amount of contact between the grazer and the phytoplankton. So you should have a relaxed grazing rate, and you should see an increased instantaneous growth rate of the phytoplankton as a result. And the mathematics behind this method are that at these growth rates, the slope represents a mortality rate on that host population. Usually this was done just by looking at chlorophyll at a time 0 and after an incubation. And the y-intercept represents the growth rate of the algae community in the absence of grazing. So what we thought, we could do something even more clever. We could add another ultrafiltration step to create a parallel dilution series that removes both viruses and grazers using a 30 kilodalton filter. And you'd think that what that would mean is that this mortality rate would be higher than the grazers alone, because you're adding another source of mortality onto those phytoplankton communities. And so hypothetically, you would get this increased slope or steepness of slope for the mortality and grazer combined, or the virus and grazer combined effect compared to mortality. But when we did this in practice, we saw that sometimes, but a lot of times we didn't see that. We saw that by adding, or by removing viruses and viral pressure on the host communities, we actually saw an increased growth rate. It didn't really make sense to us. What we were thinking is that we'd obviously, by having both mortality rates gone, we'd see increased growth, and in fact, we saw a suppression of growth when we removed viruses. So again, counterintuitive. You remove viruses and the algae grow better. What's going on there? We decided we needed to look at this directly, and so we used some classic C14 incubation experiments. I partnered with some folks at Fisheries and Oceans Canada. We'd used radioactive bicarbonate to label phytoplankton and get a radioactive measure of their growth. This is a common method, the C14 method. And what we saw there is that in these dilution series, if we just look at the picoplankton, we did size fraction and primary production. I won't get into all the details of that. But the 100% primary production value, easy to estimate. And now if we look at what the predicted value of primary production should be just based on dilution alone, so you have only 70% of your community, you should get 70% of your primary production. And our measured values were always lower than that. So it's actually showing us that by taking viruses out of the system, we were compromising the growth of the algae, not actually allowing them to grow better. And maybe going back to thinking about food web dynamics, that makes a lot of sense because we're looking at whole communities of viruses. And so by taking viruses out, we're probably short-circuiting some of the nutrient flow and some of the recycling of nutrients and materials. And that is leading to a compromised state for the phytoplankton where they don't grow as well. So it gave us this idea that viruses could actually stimulate primary production rather than just compromise it. And that was an interesting side note in the studies. Just to bring us up to the 2000s, I did have a student, Christine Palermo, I just want to mention this very quickly, who did some metagenomics on Hamilton Harbor. We had a set of samples collected at 2 different sites, near shore and mid-harbor. These are only separated by about 3 kilometers. Hamilton Harbor is an embayment at the end of Lake Ontario. And what we saw was really interesting, that the community composition shown by these pie charts at the level of large groups, so we have caudavirales, mimiviruses, phycotoviruses, so on. The nearshore samples seemed kind of homogeneous throughout the year, where 3 kilometers away, things were much, much more dynamic. And the one thing that surprised us, and maybe Thomas would be interested in this, is that the diversity of what we were detecting at that time as mimivirus-like viruses was incredible. There was all kinds of diverse mimiviruses in these samples as represented by these colored blocks. And we also discovered that there was a lot of virophage. That time they were fairly new. These are viruses that I'll get into in a second that can parasitize other viruses. And we saw a limited diversity of that, but this is clearly an observer effect because I think at the time there was maybe a handful, 5 to 10 sequences of virophages actually in the databases. So, you know, your bait for doing your bioinformatics is a pretty limited bait set. Nonetheless, we saw incredible diversity and really dynamic communities, even in a small embayment in samples separated by just a few kilometers. So we were surprised, again, that there was not more homogeneity in the samples. Okay, so that's a lot of background. Now I want to get into the stars of the show, for me anyways, the workhorses of my lab now. And that was this Chrysochromulina parva. And a long goal from starting my lab was to actually try and establish a novel model system to study host virus dynamics of an aquatic algae. And that we tried some early attempts of screening chlorella cells and things like that and weren't very successful with that. So we went back to our clone libraries, and fortunately enough, in one study we had seen this one sequence, this sequence in light blue here, which is some kind of primnesiovirus. This was from the UTM stormwater pond, where this gene sequence, again, this is DNA polymerase, a marker for these viruses, and we detected this one sequence. The rest of the time, we'd just get prosinoviruses and chloroviruses, kind of what we expected, but once we saw this sequence. And this was really interesting to me because it's a closely related sequence to viruses here, this PGV, that's Phaeocystis globosa virus, or CBV, that's Chrysochromulina baraviphyllum virus, and we saw a sequence closely related to those marine viruses, suggesting that there was probably a host in our freshwater environment that was similar to the hosts of those freshwater algae. So I went back to phycological records, and you don't have to look too closely at this. I wanted to see what others had seen through classic techniques. So Mohideen Munawar, a DFO researcher, had been studying algae in the Great Lakes for decades and cataloging them microscopically, not genomically or anything like that. And he'd have tables like this that show the species that he'd detect in Lake Ontario in different times of the year, spring, summer, fall. And one that popped out at me was this chrysophyte. Now that's the wrong group. Taxonomy's changed and moved on. These are no longer chrysophytes. These are now considered primnesiophytes, or they are primnesiophytes. And we saw this sequence, Chrysochromina parva, which jumped out at me because, again, looking at this thing over here, the CBV, that's Chrysochromulina breviphyllum. So that's a congener. That's a marine congener of Chrysochromulina parva. I thought, okay, well, we got to get a culture of parva and see if we can find a virus that infects it. It was my one great epiphany in science, I guess. So we tried to get C. parva from a culture collection in Japan. The gorillas at FedEx sent me a castanet instead of a thermos. The sample I got was like busted and had leaked out and was nothing. So then I scoured others. And luckily enough, I found that the National Center for Marine Algae and Microbiota in Maine had this culture of this freshwater species, Chrysochromulina parva, CCMP 291. So great, I got a culture of it. It's a beautiful little haptophyte cell that's about 5 microns in diameter, tiny. It has two flagella and 1 haptonema, which sometimes you can see all coiled up like this. And we started to grow that in the lab. And then we started to screen samples to see if those samples could kill our beautiful little algae. We're really nice people. So we screened a couple hundred samples. And this is just showing a plot from some of our screenings where we have a control culture just in medium. And then we also have a culture where we added a 0.45 filtrate from an environmental sample. And we got this effect from one sample from the Bay of Quinte. Here we're just measuring relative fluorescence or red fluorescence coming from the chlorophyll A pigment of the algae, easy way to monitor their growth. And we saw this effect, that this one sample causes suppression of growth, as inferred by the drop in fluorescence. And that gives you a sense that it's maybe viral, maybe biological, but not quite. So we took that material, we autoclaved and sterilized it, and saw that we would get just normal growth. But the real money shot was this. Doing the EM of cells halfway through an infection curve and seeing cells like this chocked full of virus-like particles. And so we thought, we've got it. We got the virus. We have the model system. That was a long day in the EM, but a very happy day at the EM. And we decided we would go back to our, so this is jumping ahead, 2015. We were not so metagenomically acquainted at that time. We were still relying on our old school PCR and Sanger sequencing. So we relied on our universal primers to see if we could determine if these were true algal viruses or viruses. We amplified the DNA polymerase gene, and we got dozens and dozens of clones, just a single gene sequence that we then called CPV-BQ1 for Chrysochromulina parvovirus Bay of Quinte 1. And we thought, great, we have a single virus. We were totally wrong, but that's what we thought. I put the megavirus Mimiviridae, which is now in a new classification scheme in the order Emitter-Virales. ICTV is finally starting to get its stuff together. So these are all Emitter-Virales, and these would all be algal-Virales. And there's sister groups of viruses that can be related to each other through shared genes, like their packaging ATPase or that DNA polymerase and things like that. We had no evidence that our virus was in this family at all, although we saw a lot of major capsid proteins that we also cloned and sequenced that were related to these things. And I just wanted to highlight this because there are a group at this time that we were starting to understand that there was groups of viruses, all these ones here, that actually infected different kinds of eukaryotic algae that were closely related to mimivirus. And if you're not acquainted with the virus, mimiviruses are viruses famous for being named because they mimic bacteria. They were thought to be bacteria because they're so large. Turns out they weren't, they're viruses, but they all infect amoeba. So heterotrophic protists that you've all seen in high school and in undergrad labs. But we didn't have any evidence for that, so we thought we had a virus. One of my students who was doing her master's left for 2 weeks and said, I'm going to do an art project. I was like, what? art project, but she came back with this beautiful drawing. These are false colored infected cells that she overlaid with these light microscopy derived pictures. And it turned out not to be such a bad use of her time, because it ended up on a journal cover in this book, Viruses and Microbes, which has a chapter on the diversity of algal viruses that's totally wrong, because the taxonomy was all current as of like 2005 or 2008, and it's all changed now. But the point is actually the chapter still has some good insights, because we knew a lot about the viruses at that time. They just weren't labeled properly at that time. Anyways, jumping ahead, I wanted to then go back to that virus. So we prepped the virus, we grew up a lot of it, extracted all the DNA, and partnered with a colleague, Steve Wilhelm, guy I already mentioned, viral shunt guy in University of Tennessee, Knoxville, to see if we could sequence the genome of this. And his group, the sequences are messy, because these cultures, not only will they not grow on solid media, they will not grow azenically, meaning there's a rich bacterial community and even phage community coexisting in all of our cultures all the time, which makes their study a little bit tough, but not intractable. Nonetheless, we sent off our DNA, we assembled a genome, and we got this thing, this 437 kb assembly. It's mapped as a circle. That doesn't imply that it's actually a circular genome. It was just for convenience to draw it. But a very large genome, of which at that time we could identify about a third of the open reading frames. It has almost 500 open reading frames or putative proteins encoded, and only a third of those are recognizable. The rest are hypothetical proteins or duff proteins that we don't know what they're doing. Point is, we got this new giant virus, and the DNA polymerase sequence that's encoded in this genome didn't match BQ1. It was something totally different. So it told us we had a different virus. So we called it really cleverly BQ2, and we got BQ2. But more important than that was the fact that in the assembly, we also returned several small 20 to 24 kb genomes of these things that we've called CPVV, Larry, Curly, and Moe, after the three stooges. Larry, Curly, and Moe are putative virophages, or at least that's what we thought at the time. So these are viruses that would co-parasitize a host cell along with a giant helper virus. In some ways, these are a class of satellite virus that has been seen before, but there's a little bit of nuance to that. And let me just jump into that for a second. So the first virophage discovered was a virus that infects amoeba along with mimivirus. And it was being recognized as a satellite virus called Sputnik. And this is what the story of Sputnik. If you have the giant virus, the mimivirus, infecting amoeba alone, it sets up its viral factory and it makes a lot of progeny. Great. Now, when you add Sputnik and the mimivirus into the culture together and grow them, you see that Sputnik will compromise the growth of the mimivirus, leading to that term virophage. It's actually parasitizing the viral factory of the giant virus. The first reports of this had viruses inside of viruses and all kinds of silliness like that. That's not true at all. But the point is that the satellite virus co-infects and compromises the fitness of the main virus. And there's even one special case of a virus that infects Cafeteria robergensis, a heterotrophic nanoflagellate, that virus, Ma virus, actually resides within the genome inserted in the giant virus itself. And so with one of these latent infections of a virophage in a giant virus, you can infect with that giant virus and get both the virophage and virus being produced. So there's a lot of dynamics potentially happening in these tripartite systems. So knowing that we had two viruses, this is now just, in the last few years, we've really tried to tease apart these viruses and get them growing in pure culture. For years, we tried to do one-step growth curves, I was telling the group at lunch, and always failed, thinking that we were just dumb or losers, we can't do it, whatever. But turns out you can't do a one-step growth curve if you have a cocktail of viruses. It just doesn't make any sense. And now that we know that we had a cocktail of viruses, like, oh, okay, good. We don't have to panic. So we decided we could grow these viruses and try and tease them apart by doing our endpoint dilution assays. I'll just show here the one thing that we do know is that this is a control culture. This is if we take a viral lysate and filter it through a 0.1 filter, we can remove all infectivity. But if we pass that same lysate through a 0.2 filter, we get true lysis of the cells. But by growing these things in a 96-well plate like this and isolating viruses from the lowest dilution that lysed, kind of like a plaque assay, we could purify these things. And so that's what we've been trying to do over the last couple of years. I'm going to just hint at something here. We have all of our qPCR methods that we still use to track these individual viruses, not just in the environment, but in cultures. So we have assays for some of the viruses that infect the host, as well as these virophages, so that we can track them all together. We've done EM to prove that these are viruses, and the EM that we use in this this thermo down in the medical imaging labs in University of Toronto. The top panel represents the BQ2 virus, which looks like a mimivirus in that it seems to have this fibrous sort of integument on the outside of the capsid, and they're huge. You can see they're, in the 250 to 300 nanometer range in diameter. And in this one, this little particle here, we think it is Mo. And I'll come back in a second. This is the second panel. These are the BQ1 viruses, much smaller, but looking like a true phycotoviruses. And on the bottom panel is preps of Mo, purified preps of Mo. And I'd say that this picture right here, this negative stain image of Mo, is my favorite virus picture I've ever taken, because Mo looks just beautiful there. And it's a tiny 70 nanometer particle that co-parasitizes, as near as we can tell, along with this virus. And that's the story for the last thing. One thing that we always get asked is, do any of these viruses infect the host in a latent manner? So when asking about how do the viruses persist over times when host abundance drops below detectable limits, where you'd expect transmission to be occurring, how does the virus last in the environment when you show that it can be destroyed in days in the summertime? So it must be residing in the host. But every time we interrogate a naive culture of the host that hasn't been infected with viruses, we can show that we can easily amplify, eukaryotic genes from it, but we never detect any viral genes. And on the other hand, when we take a lysate of that culture that we've infected and we purify that lysate, we can show that we can remove all the host genes, but we always detect all of our viruses there. So we know that we suspect that it's not a latent infection that's supporting the growth of these viruses over time. Last summer, we decided we'd just see what would happen. This was Ichiro Inamoto, my lab tech who works part-time in the lab, just tracking a culture if we transferred one lysate to the next each time, looking at it with qPCR. And after 8 generations, we were really shocked that every time we did this, the lytic effect was maintained, meaning this lysate that we were generating could kill the culture of the hosts. But we saw that the 2 CPVs we knew of, BQ1 and BQ2, disappeared, and we couldn't detect them anymore. But Mo was really, really highly abundant. So that made us question, is Mo actually a virophage, or is it able to infect on its own, or are we missing something? Are there other viruses there? So knowing that we could remove infectivity with a 0.1 filtrate, we filtered a lysate through a 0.1 filter. We show that that's not infectious. Mo remains highly abundant. It should. It's only 70 nanometers. It should pass through that filter, and it does. But none of the other viruses are detected in this filtrate material. However, if we take that same filter and rinse it off into a culture, we can show that it is infectious. And moreover, if we add more Mo to that material, we see an increase in production of Mo. So it seems like whatever is on the membrane is infectious. It's not BQ1 or 2, and it supports the replication of Mo. So back to the genomics bench, and we assemble this genome. We haven't published this yet. It's a partial assembly. We're still trying to work out some things. The contigs are not matching well, but it scaffolds really well onto BQ2. It seems like it's a relative of BQ2. The Paul gene is 93% identical to BQ2. Being as clever and ingenious as we are, we call it BQ3. But one thing that was really gratifying about this underway genomics project is we returned 100% identical genome assembly of Mo. And if you look at this was Illumina sequence, that's 50,000-fold coverage, which is ridiculous. Easy because it's such a small genome, but we're really confident that we have the true genome sequence of Mo. It's inscrutable, because this was different DNA preps from different lysates done with different sequencing and using different bioinformatic approaches. And when you get the exact same thing back with all those differences, you're kind of happy about it. And recently, we've actually been able to go back to BQ1, that now we have it growing in pure culture, and get the genome of that. So I partnered with Trevor Charles and Joseph Nizamov in University of Waterloo, and their student Delaney Nash, who was at the CSM meeting. Some of you might have met her. Brilliant student, she used a nanopore long-read sequencing to create a scaffold of this genome because it doesn't really match anything else that we could use. And then error corrected using Illumina sequencing. And she got a very high confidence assembly of the CPV BQ1 genome, which is past preprint. This is now published in BMC Genomics. But we're really happy about this. And like all of these giant algoviruses, it's not so giant. It's maybe mid-size, 165 kb genome. But it encodes all kinds of wild things that we don't really expect to see in a virus. Methyltransferases, restriction modification systems, which we originally thought were maybe due to or for the purpose of recycling host nucleotides and things like that in replication, but maybe it's more about competition between these two viruses competing for the same host. Those are questions for another day. Okay, so here's where we stand right now. We have that we know of, and there's more because we have things in the lab that we're still trying to characterize that don't fit these. But we have 2 of these algovirales, BQ1 and BQ4, and 2 of the emitter virales, BQ2 and 3. These are the giant viruses with the 380 kb genomes, or sorry, 480 kb genomes, and these are the smaller, what would have been called phycotonaviruses a long time ago. And BQ2 has actually been formally classified as Tethys ontariense by Frank Ioward when he revisited the whole taxonomy of the emitter virales. So we have 4 viruses to play with and lots of things we can do. Now, we want to go back to that question of what's Mo? Is Mo truly a virophage or pollenton-like virus or pollentovirus? And we decided to conduct this experiment. This was 2 students of mine, most recently George Thomas, who worked on this as a senior thesis project. We take naive C. parva cells. We have our controls in triplicate. We have a treatment where we add just BQ3 alone, a treatment where we add just Mo alone, and a treatment where we combine the 2 of them to see if Mo can replicate on its own or not. And the results are interesting, and actually we're learning more that the ratio of Mo to BQ3 matters. And I'll just say that for all of these infections, BQ3 was held at a very low MOI of 0.01 infectious units per cell. But that's enough to cause the collapse of the culture. As you can see, the yellow line is the control that grows very nicely. The black line is the BQ3 infection, which completely compromises the growth of the host, even after 5 days at that low MOI. When we add Mo at alone, nothing happens. Mo doesn't replicate, Mo doesn't lyse the cells. So that confirms its kind of maybe satellite virus status as a virus dependent on a helper virus for its replication. BQ3 at low at low concentrations relative to BQ3, a 1 to 10 proportion, does not protect the cells at all. Maybe a little bit at the end where we saw some growth recovery before the culture collapses. These are batch cultures, so they don't keep going forever. But at a high concentration of Mo to BQ3, so when we add when we add Mo at 1,000 to 1 to BQ3, we see that it completely protects the growth of the host cell, which can keep growing in the presence of BQ3. So a high enough proportion of Mo can actually stop the lytic effect of BQ3. Question is Mo being replicated and all that? So we've, of course, done our qPCR stories with this and looked at it. So this is now, on this panel, we're looking at qPCR of the helper virus, the BQ3 virus. And you can see that in the infection of BQ3 alone, we get rapid increase of BQ3, so it's replicating as we'd expect. Mo and BQ3 at low abundance does nothing to BQ3's replication. It replicates quite happily. But in the presence of high concentrations of Mo, again at 1 to 1,000, we see that the BQ3 replication is compromised quite a bit, and we don't get the replication of BQ3 as we want. So Mo does seem to be having that virophage-like detrimental effect on its helper virus replication. When we turn tables and look at Mo itself, we can see that Mo is being produced both at low concentration. We see a rise, even though it's not protecting the host cells or compromising BQ3's replication, it itself is replicating very well and increasing in abundance. And at high concentrations of Mo, of course, we lose a bit of signal because we're starting with so much Mo, but we still see some evidence of Mo replicating, even though it is actually compromising the growth of BQ3, its helper virus, and shutting down that infection. But it still seems to have some action. And Mo alone does nothing, so proving that it is a dependent satellite virus that requires its helper virus. And I'll just say that in the gray notes are that in these treatments, we never see anything we don't expect, meaning viruses that shouldn't be there. They're not there. We don't detect them. Okay, and the last little story I'll tell you about is something that's really interesting me right now and kind of a focus in the lab trying to understand how this is happening. I was at a virus persistence workshop in a beautiful place in Portugal. That's besides the point. Many years ago, 10 years ago. And when we were talking about viruses and how they replicate in the environment, a few of the modelers and physicists said that there's no way a virus could actually replicate and cause the extinction of its host culture, because that's a dead end. That's an evolutionary dead end that shouldn't exist. And I said, well, when we add our viruses to our host culture, the host culture collapsed to absolute death. There's never any recovered cells. There's never cells that we can regrow. So we had lysates sitting in our fridge and in our incubators that we transferred into new media for 6 months, years, and we never saw any cell regrowth. So it seemed like there was no evidence that these cells could ever be resistant to infection. Well, that was when we were so naive as to think that we had one virus. Now, knowing that we had multiple viruses and cocktails of virus, maybe that makes sense, because now that we've purified viruses and are growing independently, we're starting to see all kinds of abilities of the cell to grow in the presence of viruses. So this was Ichiro and another fourth-year undergrad, Heiji Kim, did this work where we looked at just following a culture over generations, where we saw that the culture would grow well, we dilute it with fresh media, continue to grow, dilute it with fresh media, and continue to grow. But at the same time, when we were following that culture with qPCR, looking at what BQ1 was doing, we saw that BQ1 gene copy abundance, maybe not viruses, we don't know at that point, but gene copies at least, were increasing and then diluting down with our new media and increasing. So what this looks like to us is that the virus, the CPV BQ1 virus, is coexisting with its host cell. So the hosts are growing in the presence of an infectious virus, and then infectious virus is not a dud and it's not complete resistance because it's continuing to grow as well. And that becomes an interesting phenomenon to try and understand. So Heiji did this, again, simple experiment, growing our cultures. Now this is the BQ1R or BQ1 recovered culture. I'll say that that's this guy here that we're growing up. And she did the treatments of adding BQ1 in duplicate, unfortunately, and BQ3 in duplicate, BQ4, and then just a control. And what we were trying to see is what was the range of this so-called resistance or ability to resist infection. And we collect samples every few days for a month through the batch culture growth. We do qPCR for our viruses, and we just look at a hemocytometer and light microscopy to count cells. And this is what Heiji saw in that experiment, that the host cells, the black line is the control culture, just growing kind of normally. BQ1, the host cells continue to grow okay, so that's in the presence of BQ1. In the presence of BQ4, they also grow quite well. That wasn't that surprising, because you remember back to that phylogenetic tree I showed, BQ1 and BQ4 are very closely related. So it seems like whatever mechanism is at play to support the resistance to BQ1 also works for BQ4. But then crossing over into the other order, into the Immeritavalles, that BQ1R culture cannot withstand an infection by BQ3, which causes lysis as always. So it seems like it's at least a mechanism that's specific at the level of the order of the virus, not beyond that. I want to redo this experiment though and have it properly in triplicate so we could do some statistics and really demonstrate what's happening here. In this case, now we've been growing this for a long time, and I can say that we have stably in lab a culture of C. parva that just produces BQ1 gene copies constantly and grows just happily, surprisingly, without really any detectable difference in the growth rate of a naive culture versus this BQ1R. And that's something I don't really understand at all. But nonetheless, we did this experiment with BQ1R, again, just repeating it with BQ4, BQ3, and our no infection control. Just to give you an idea, we just set these up as flasks in our incubator. This is looking at how the experiment proceeds over time. We make sure that we know the light field and things like that, so we're not getting artifacts from the growth condition of the cells. Kind of trivial point. But again, we get the same effect when we did this. Our control grows fine. In the presence of BQ4, there's no effect on the host cells. They continue to grow. In this case, BQ3 caused an immediate lysis, but some of the cells recovered and then sort of pooped out again near the end of the growth. We've saved this culture. We're going to start to investigate that now, see if it is a resistance to BQ3 that's emerged or not. And then this is really the summary of what that experiment showed. And that is, if we look at change in gene copies for each of our virus, so the colors represent the virus we're tracking with qPCR, and on the bottom, each set is the different experimental treatment, right? So when we add BQ3 to the host culture, we see that our BQ1 viruses drop millionfold, and they're not being replicated at all. So BQ3 causing the lysis of the culture also blocks, not surprisingly, reproduction of the BQ1 virus. The BQ3 virus, again, not surprisingly, increases greatly. 10 million, 100 million fold increase in its gene copies over the course of that growth. And on the other hand, we didn't add BQ4, so nicely, it's not detected. When we look at what happened with the BQ4 treatment, here we see that BQ1 is still being produced by that culture, but BQ4 is also being produced at the same time. So it's not specific to one or the other of these viruses. It seems like it's an additive effect. And BQ3, of course, wasn't added, so it's not there. And we look at our control, again, we see BQ1 is being produced by this control culture all the time, and we don't detect any BQ3 or BQ4. And the interesting thing about this is BQ1 alone is about a twofold increase in the change in gene copies over the experiment compared to the combination of BQ4 and 1. So it looks like when BQ4 is there, half of the resources are being shunted into BQ4 production, so it's reducing a little bit of the BQ1 production. But the point is that this culture is a stable thing that's able to grow very well in the presence of BQ4. And it's a little bit puzzling because it just keeps growing and growing and growing and doing this all the time. And we don't really know why. So looking back at the literature, last little thing I'll say is that it gets complicated when we're dealing with eukaryotes. These cells, as far as we know, there's evidence that they have sexual reproduction. So when you look at the C. parva genome itself, it encodes a lot of the genes for meiosis, suggesting that it does change its state from diploid to haploid. Now, the close relatives of this, marine coccolithophorids, a species that used to be called Aemiliani huxleyi, there was this observation from years ago that showed the diploid cells could be infected by viruses, but the haploid cells were absolutely resistant. And what these researchers were saying is if you take a diploid culture, in this case, EHUX is easy, because the haploid and diploid cells look different. You can actually see them. The haploid cells are flagellated, naked cells. The diploids are encrusted with coccolithophorids. So you can kind of, or coccoliths, sorry. So you can tell them apart. And the point is what they saw is that the viruses infect the diploid phase, and in a virus infection, it triggers the cells to go into meiosis, to give rise to a haploid resistant state that cannot be infected by those viruses. And they call this the Cheshire cat after Alice through the looking glass. You know, the Cheshire cat cut its head off so that the queen, I can't remember the story. But the story is here that we have a phase change or a ploidy change that protects the cells from resistance. And now there's another study from a great research group in Israel. And they've shown that in the same Aemiliani huxleyi system, it's funny, they didn't come back to thinking about this, but they've shown cell-to-cell heterogeneity in this population, meaning you can take a single cell that's resistant and grows in the presence of virus and subculture that, and some of the cultures are susceptible to infection and some are resistant to infection. So it means a susceptible culture or a culture itself is a composite of both susceptible and resistant cells, the susceptible cells allowing the generation of viruses, and the resistant cells allowing the propagation of the host cells themselves. Whether this is also due to a ploidy switch or not is not clear in that study, but they did single cell work on this. And I think this might be the same for C. parva. C. parva, dealing with eukaryotes, the sexual reproduction of it, like I say, is not characterized, it's not known. And if there is different diploid and haploid phases, they're isomorphic, so we can't tell them apart. So we're trying to figure that out right now, because one intriguing idea is, again, the viruses infect only one phase of the growth. And so viral infection actually can stimulate and trigger sexual reproduction. And what would be really amazing to us is if the emitter virales infected one ploidy, And the phycotoviruses infected a different ploidy, allowing a system that could sustainably produce both kind of viruses depending on the ecological context. So that's all I want to talk about. The summary here is that we know that there's lots of diverse viruses in these populations. We've been able to isolate multiple viruses that infect this one host. Again, that's what I call this ecological churn. Every time we look more closely, there's more there. And so this is a single algae out of the potentially 40,000 species on Earth. This is one algae, and it has a whole cocktail of viruses attacking it all the time, and yet it grows stably in its environments. We now know that Mo is truly a virophage and behaves in that way. We're still working on Larry and Curly, and it doesn't seem like it's quite the same for Larry and Curly. They might be partnering with one of the other viruses, which is also something we'll be following up. And we know that we have this productive replication strategy, which is kind of unusual and maybe due to some physiological characteristics of the cells themselves. It is not a genotypic, it seems like it's a phenotypic shift, and you have a population of susceptible and resistant cells coexisting. And of course, I have to thank all the people. I know I went a little over time, apologies for that. So I'll just acknowledge all the people that had a role in this. Those are some of them, and leave you with this. This is generative AID's picture of giant viruses in an aquatic environment. And some of them are really into motorboating, apparently with large wakes behind. So thanks for your attention. Happy to take any questions.